Okay.
So next March, we're going to be switching up our presentation date to the 20th to accommodate Bo and I's schedules, but we hope that you can all make it to listen to Alex Gatch talk about transport stress limiting success of bloater reintroduction in Lake Ontario. And I will be dropping the link to register for that in the chat.
Thank you very much, Renee, and thanks to Alex for recently agreeing to give that presentation next month. But right now, I have the distinct pleasure to thank Bill Carter for jumping in very relatively last minute and offering to give what I'm excited to hear is a follow-up talk to Randy Brown's webinar that Randy gave to us last month about Inconnu. Let's see, Bill, you can share your slide deck if you'd like while I introduce you. So Bill has a very interesting background as well. He grew up actually on the east coast of Florida, where he enjoyed uh, beginning to interact with nature in the tide pools and being a lifeguard on the beaches. He came to Northland College in Wisconsin, which many of us know in the Great Lakes, on Lake Superior, to play basketball all the way from the east coast of Florida. There at Northland, he majored in biology and organic chemistry. He ended up in Alaska due to connections for running sled dogs and being a factory trawler observer, and ended up staying out there, becoming a fisheries field technician in several of the throughout Alaska and Denali and other places, and ended up getting his master's in fisheries at the University of Alaska in Fairbanks in the early 2000s. Since 2014, he has been a fisheries biologist at the Selawik National Wildlife Refuge for the Fish and Wildlife Service. And this is above the Arctic Circle where he lives. And because I think it's so fascinating to kind of more about the geography of where he lives and works, he's just put in a few slides to give us a little bit of background about that refuge before he talks about his broad whitefish work. I guess the last thing I'll say is the broad whitefish work is from his master's back at Alaska Fairbanks in the early 2000s. Um, he thinks the talk will be about 20-ish minutes, maybe longer if he gets to telling some stories. But essentially, he's retelling his master's work from more than 20 years ago. So thanks again for stepping up, Bill. And we look forward to hearing your presentation.
Yeah. So Bo asked me to drop a couple of slides in. I happen to have them from from another presentation, but they're in a slightly different format. That's why you can't see some of the things. But just to give you an idea of how big Alaska is to begin with, it covers up pretty much 10 of the Midwestern states there. The light colored insets of the in the Alaska map there are the 16 refuges that are within the state of Alaska. Some of them have been complex, but there were 16 distinct refuges when Anilka, which is the founding of the majority of the public lands in Alaska, were kind of codified. So, we have Alaska has the easternmost and westernmost point in the U.S. because the 180th parallel goes between the Aleutian Islands out here. So we have some distinct or some unique geographic spots, and that's the refuge that we're going to talk about here, or where I work currently. So another quick thing here. So the Arctic Circle, you can see the Arctic Circle pretty much runs right through the middle of the refuge here. We have some wilderness land, and we have a wild and scenic river corridor in the upper Selawik. The refuge is 2.15 million acres. We have some native land, this darker colored stuff around the village of Selawik and the village of Norvik, and up here in Kayana. That's due to the way that Anilka is structured, there were private lands that were ceded to village and regional native corporations. And Anilka all has to do with the land claims because we don't have any reservations here for our Native Alaskans. So they got corporations instead, for-profit corporations rather than land. And then we have subsistence rights on federal lands for those native. Well, it's rural preference, so I even have subsistence rights. So, bunch of trade-offs there, but it's a unique and complicated situation. So, anyhow, so moving on to my master's work. Some of this will be rehashed if you saw Randy's presentation, but it's definitely a unique fish. So Alaska still is pretty untouched if it's not a commercially viable fish. There's not a whole lot of money for it, so there's lots of work done on salmon. And other than that, you can still do life history studies even today. And that's what this is basically a life history study because we just didn't have enough information. So we had some length weight data from a few people like Ken Alt that Randy spoke about in his presentation. We had a few distribution records, and then we knew that they also had they spawned in the fall like the other coregonids. So the rapids is a really unique spot. It has a distinct gradient to it. If you're here on this fish wheel, which is where we did all our captures, you can actually see the gradient between the upstream and downstream of this place called Rock Island. It's a hard granite dike. We're actually going to use it to build a dam for the basis of a dam, but it splits the river. The river's about a quarter mile wide here. It splits the river into two channels. At this water level, probably about 30 feet deep on either side, but because of the current, it pushes the fish that are migrating upstream here off to the sides, which makes the fish wheels really effective capture methods. So it's 120 kilometers upstream from the Bering Sea, and we chose this site, one, because the fish wheels were available, but two, it's far enough up from the Tanana River that we were pretty confident that the fish migrating past here were going to continue up and not be. It would have been more convenient to be down by the mouth of the Tanana because there's a village there, but this way we have pretty good confidence that the fish are going to continue upstream from here. This was like I said before, if you don't have money for something, you don't get things done. So it was a salmon project to start with, and they noticed that there were a lot of whitefish being caught. Randy first did his work on Inkanu or sheefish, and then I followed up. So we'll take a look at the 6 species of whitefish that were captured here, and then the fish wheel. So I thought I'd throw this video in here because you guys probably have never seen what a fish wheel is. It's basically a current-driven mechanical dip net. So the current pushes on either the baskets or the paddle, and that's what this thing flopping around up here is a paddle. It's actually pretty windy this day, so that's why it's stalling a little bit. Each fish wheel is designed and tuned specifically for its site. There's fish wheels that are as small as something you can tow behind a pickup truck to this monster that has 20-foot deep baskets and 6-foot high sides and stuff like that. So there's a fish there was a fish cut. Anyhow, so this part right here is a lead, and it pushes fish off the bank, and it goes underneath the raft, and then it scoops up. If any of you guys are Yukon Man fans, this is Stan Zaray. He's the owner of the fish wheel that we rented from. So he's always out there tinkering, trying to keep this fish wheel moving for us. Just another shot of it from up close. There's a slanted padded chute because this is a fish-friendly fish wheel, as Stan says. But then once it goes down this chute, goes into this chute, and we have a camera system with lights so we can count these fish. And that's how we got our CPU E that in this project. So just keep spinning around day and night unless it's too cold, and then we have to stop it so it doesn't freeze up. This quick still is just the other side. This is a special fish wheel that has a trigger mechanism here to run the camera. So that would collect frames previous to the trigger, so that we didn't have to watch a bunch of empty video of no fish going by. So that's a whole nother presentation in itself. So all these fish were caught pretty much, they're obviously caught in the same day because they're all nice and fresh here. But when Randy worked on did his ground truthing up on the Yukon there, he found the humpback whitefish and the Bering Cisco and a few least Cisco in the same spawning area as the she-fish, but he didn't find any broad whitefish. So, and round whitefish are really very scarce. I don't think I saw more than a handful each year. They're usually just up in the high tributaries. They don't usually migrate through the Yukon very often. So, so yes, I decided that I needed a grad project and was granted to work on broad whitefish. So it's a real robust fish. Mostly eats mollusks, snails and clams. And Randy, the godfather of whitefish, also proved that broad whitefish are a vector for moving mollusks around, because he found out that the mollusk can pass all the way through their intestine and come out alive on the other side. They've got a real muscular stomach that they use to grind up those shells, and some of them don't get ground up, so they move those mollusks around. So pretty straightforward life history. We want to know if the fish are spawning. Want to know how, what the timing is, how long it takes them to the whole run to move through the point here. We use radio telemetry to look for their spawning area. We used a thin section otoliths to age them. Found that scales don't work really well on older cold water fish. We had discrepancies. Older works said that broad whitefish were maxing out at 9 and 10, and we found different results from that. So, and then microchemistry to see if they were moving down to the ocean. So we started collecting, just kind of haphazardly collecting fish to get GSI, and tried to spread them out through the whole period of the project. Started usually early August, and by September they were big masses of females. And because Stan also has a dog team, it was really easy to get rid of these fish. There's several dog teams around that area, so we can even though we're sacrificing the fish, we're feeding them to a good cause there. One of the interesting things we found was that the GSI can vary quite a bit. Our smallest and youngest fish had the highest GSI, and there's no real correlation between age or body size as to what their GSI is. Some of this is skewed a little bit depending on when they were actually caught. If they were caught earlier in the season, they're obviously going to have a smaller GSI than later in the season. So in this, the dotted line is August 15th. And we kind of arbitrarily chose that date because we were getting at least five, five or six fish a day. But you can see that they were pushing up into the 40s per day. And you may notice that we have these abrupt ends towards out at the end of the CPUE data because we'd have to shut the wheel off at night if it started to ice up. And most of the broad whitefish are moving at night, so the counts kind of fall off abruptly there. So here's our severely overqualified crew. Randy's getting ready to do a surgery on a fish. The guy with the net here is Dave Dahm, who's the guru of the camera setup, and then my advisor Gordon Haas is there. And we were using this flow-through system for anesthesia. We were using clove oil at the time. So we had 2 buckets up there on top of the cab to administer the anesthesia, and then a freshwater bucket to start bringing them out right before the surgery was finished. So we had a pretty slick setup. We'd put the fish into an induction bath. Then when they were ready to go onto the cradle, we'd tear the scale and then put them in the cradle there and do our surgeries. It's pretty quick. We could get a fish from the induction tub into the freshwater recovery tub in about 7 minutes. And part of that was because we were when I'd start this first suture here, we'd start hitting them with fresh water, and that way they would recover a little bit quicker and not be under anesthesia so long. So we found that broad whitefish and whitefish in general are kind of wimpy. So we put them in this induction or into the recovery tub just long enough until they were upright and they could, maintain their equilibrium. And then we get them out into the water so they could find a place that they could just hang out. So when we would after we released them, the next day we'd go out and we would try to figure out where they were using boat tracking just in the general area. And most of them started moving 1 or 2 days. Some of them were really wimpy and would take several days before they actually moved. But then we would use these remote towers that were, again, set up for the Chum and Chinook project by NOAA, but it gave us the opportunity to track these other fish. And then finally, we pinpointed their positions on the spawning grounds with airplane aerial surveys. So this is just a quick map of the tower setup. It basically broke up the drainage, and they're more than the ones that we used farther down, breaking up the Kayakuk and the Inoko. But this allowed us to concentrate our aerial surveys if we knew fish had gone past a certain tower, but not past another one. So this is this is the Rapids tower, and then this tower is on the Chandelar, and then and they were all far enough up that the fish weren't like milling at the confluence of one or the other rivers there. So my first year, we found that they were all clustered in this section between Fort Yukon and Stevens Village. And Randy had found all of the other whitefish up here, but the majority of the least ciscos were up here on the Chandelar. We had another project to deal with that because they weren't. But you can see that this is a much more highly braided section. The gradient's much higher here. Most of the islands there are just willows or just bare rock because they get scoured by ice breakup. Because up above here, the river narrows again into a single channel canyon-like section that can have huge blocks of ice when it breaks up. And this section, as we'll see in a couple slides, is much older islands with big spruce trees and just a little bit different habitat. So my second season, fish ended up in the same place, spread out a little bit differently, but for the most part in the same place. One fish decided to go up and play with the other whitefish up there above Fort Yukon. But what we hadn't found was Randy found that all of his Inconoo had gone back past the tower pretty quickly. By end of November, they were all back past the Rapids Tower there. But I never found any of mine, and we didn't know whether they had moved really late in the season because they were up here really late in the season. And the tower had run out of power because it's a solar powered setup, and the batteries would drain out late in the year because they didn't get enough sun, and then they'd kick back on and they'd be fine for the next summer. But we hadn't found any broad whitefish, so we went back out in April and found that all the fish from that second year were within a kilometer or two of where we found them in December. And then one very interesting thing is this is the same fish. We went back out in September of that same year to look for repeat spawners. And we didn't find any repeat spawners on the spawning ground, but we found this fish that was here in April, up here in this Chauvin Lake off the Christian River. It's a big chain of interconnected lakes that it was up there, and we have some theories about that, and that'll be discussed when I get to the microchemistry portion of this. So this is what that section looks like. Part of our so the fish are hanging out on the downstream sides of these islands, anywhere where there's a little bit of lee from the direct current. And we think part of the reason these fish are using this area is because these islands are being a heat sink. They're pretty coarse gravel, so there's flow through them all during the year. The top obviously freezes up, but there still seems to be plenty of flow, and because they warm up during the summer, they may be holding that heat and giving it off and giving optimum places for these fish to lay their eggs. And this was taken on the same day. Depends on where you are. You can see that there's open water. It's probably 10 below or more, and has been that way for several months. This is a late November, early December picture. So on to aging. Randy got his techniques, and I learned from him from geologists. So we use a polarized transmitted light, which gives you this nice black background. And the only light that you're seeing is what's being refracted by the crystals in the otolith. This one happens to be one that we mapped also, so there's a whole bunch of junk outside of it that was blasted off by the microprobe. That's why there's all this fuzziness out here. But so this fish is at least 12. There's a question on the last growth increment there. But I want you to remember this big wide growth increment between 3 and 4 and 4 and 5, because as we move along, we'll show in the microchemistry section how why we think that's true, that is important. So age at length have wild variations here. These are broadcast spawners. They're in a river system. So and they emerge during the breakup time. So they're coming out right when everything's flooded, and then go to a whole lot of different areas. Some of those areas are good during the spring. You can get washed in there, but they have no outlet. So all those fish are going to die. But then we have fish that either get washed into prime areas close to the spawning ground or may get washed all the way down to the estuary. And that's why you have like these 6 year olds, there's 21 centimeters difference between these two fish. So there's wild variation in their, and we think it's due to their young, early years feeding. So getting on to the microchemistry, this is the same otolith that I showed before there, and you can see the lighter the color, the more strontium, and that's our indicator element. The microprobe has a very limited number of elements that they can detect, but because strontium has such a wide variation between their freshwater and marine signatures, it's a really good one just to look for anadromy. So here, with the help of my media savvy wife, I managed to squish the otolith together with the chemistry map and the optical map together. And the line where these are divided is about where the microprobe went through. So that's what this graph here on the bottom is, a microprobe line from here to the core. So, and you can see here at between 4 and 5, these fish are out in, maybe full marine water at this point, or very close to it because it's very high. But you can't exactly equate water chemistry to the microprobe data, but you can see that this fish was definitely in marine influenced water at this point and the year before. So to test what the number of anadromous fish were in the population, we took randomly selected 12 males randomly selected 6 males and 6 females, and only 2 of them, 2 males there with the highlighted sex and age, turned out to be non-anadromous. All the other ones are above this level of 1,700 milligrams per kilogram, which we're currently using a different ratio or percentage to get that anadromous number. But like I mentioned there, that Chauvin Lake fish, what we think is, if you look at all of these fish that went to marine waters at some point in their life, they all come back to freshwater. So it appears that the fish are going down there, getting size, and then they're finding a new place to feed after their first or second spawning event. Most of these fish are pretty young. There's an 11 and a 13-year-old in here, but all the other ones they're the heart of the population as far as the median. It was 9. So the other kind of interesting thing is that on the top row there, that maximum 14.6, that is the highest strontium count that we've managed to get in any fish that we had sampled up to this point. And I saw Randy's on here, so if he has something that's higher than that, he can let us know later there. So wrapping this up, these are definitely fish that are on a spawning migration. Their run timing is August and runs into September. Don't know exactly how late they run because our capture method is not safe at that time. So they're using a different portion of the Yukon Flats, which is actually part of the Yukon Flats National Wildlife Refuge, encompasses all of the spawning area for both the Inconoo and broad whitefish and the Chandelar River there, so for the least Cisco. And the very different spawning area than the Inconoo spawning area. and a different lifestyle. They're staying in the spawning area all through the winter, and we're not exactly sure when they decide to move, whether it's before or after breakup. So we have fish as young as 5 that are shown to be in spawning condition. And then we have fish, the mean for both males and females is 9, and we have a 17-year-old. 80% of these fish are going to the estuary, marine influenced waters to feed. But we don't know a lot of things, and these are still questions that are out there. Juvenile movements, it's really dangerous to be out there trying to collect juvenile fish during breakup or while they're while they're hatching. We haven't established what repeat spawning might be. We have some data for sheefish and in different places, but broad whitefish don't have a whole lot of data on them. And then what these fish are doing after their initial couple of years of spawning and that movement, like into Chauvin Lake and things like that. So that's all I have at this point. Any questions?
Thank you very much, Bill. Very interesting talk, and I appreciate the detail you provided, also showing us, teaching me at least more about a fish wheel and seeing how that operates. That was pretty cool with the video there. And then a really interesting life history you described. And while people can now ask their questions via the chat, or ideally just by raising their hand and popping the camera on, Bill has agreed to stay, for 15 or 20 minutes, or as long as people are interested in having a conversation about his research. First one from Nicole Watson. Do you have any insight on potential impacts of the recent typhoons on, I guess now current coregonine populations in Alaska?
So that mostly affected the Kuskokwim, would have affected the Kuskokwim Delta, which is Bethel area, and I'd have to say that unless there was a fuel spill or something, that's probably not a big issue, because these fish can handle marine influenced water. So if the storm surge had moved in there and pushed a bunch of marine water up there, they can deal with that. Our other our southern fisheries office here in Fairbanks, in Fairbanks, they're in Kenai. They work on the Kuskokwim. There's a big division of the state. So the fisheries office in Fairbanks is Yukon Drainage and North, and the Anchorage Kenai office is Kuskokwim River and South.
Okay. Peter Cron, would you like to? Yeah.
Sure. Yeah, my name's Peter Cron. I'm in North Vancouver. First of all, I just really appreciate these slides. They're very relevant to what I'm doing. I just sort of want to let you know that I've developed and patented a new selective fishing system. So the fish wheel aspect was really of interest to me. We've used this system here for tests in the Fraser River to catch salmonids. And in April through August, I'm going to be using it in Alberta to go after invasive species. And of course, one of the things we're going to be sampling is otoliths and all that sort of stuff. So this is very relevant because I'm recruiting some students, and just want to thank you for this, because it's very good illustration. So appreciate your study. It gives us some perspective of what we'll be doing in Alberta.
Sure.
Fantastic. So, Bill, just thinking about the life history you described, do you think that all fish are going out there, and most fish are going out to the estuary to sort of, as you said, fatten up and then starting their spawning? Or do you think any of the spawning occurs without fattening up in the estuary?
Well, there were those two males. I mean, at the time it was a little cost prohibitive to do a huge sample of microprobe stuff, but um, so yeah, there were those two males that hadn't gone to the estuaries. So and there are a lot of different possible spawning grounds that haven't been examined at all. So there's the Anoka River is in within the Anoka Refuge. It has Inconnu spawning definitely, but I've been in there netting for other things, and we always caught humpback whitefish and broad whitefish and least ciscos. Um, so there's other possibilities and other life histories. These fish are, morphologically and habitually plastic. They can just kind of change to do whatever. We have all of these fish here on the Selwick Refuge also, and again, we don't have money to do a whole lot besides find out where their spawning ground is, but they're super important subsistence fish. So yeah, it's the name of the refuge and the village is Place of Sheefish. Selivik is Inupiaq for Place of Sheefish. So
Yeah. And then broad whitefish looked like it was maybe the second largest of the whitefishes. I don't know if
yep,
that was just that's representative of,
yep,
that's a general. Okay.
yeah.
Um, and I mean, how did management sort of use the information about, like, has the location of the spawning been used also for management, or is that just more for
A little
general
bit.
knowledge? Yeah.
A lot of these villages don't have um, well, they need gravel for roads and things like that. There are no paved roads in most of these villages and stuff. So their easiest uh, source of gravel is just to get it out of the river. And some of that is, they regulate it both through the state. Another ANILCA issue is the state owns submerged lands on navigable waters. So the state permits those gravel mining things, and we've encouraged them through these studies to uh, not do it, try to do it in the middle of the summer, not, in the spawning season, right?
Yeah.
Because sometimes they like to stockpile things right at the end of the season so that they can have things early in the. So anyhow, yeah. So that's about the only management. And like I said, the the whole section of the river there is within the boundaries of the Yukon Flats, but similar to the Selwick Refuge, each one of those villages has private land, which is either village corporation or their regional corporation land surrounding them. So there's private land within refuges here, but the
Yeah.
refuges are gigantic. I want to say that the Yukon Flats is, pushing 8 or 10 million acres.
Kevin Fraley left a message in the chat. Um, hey, Bill, if you had funding and time, what would you want to study next with broad whitefish to build upon your work you just shared with us?
Well, in the Yukon drainage, it would probably be mapping out as many spawning areas as possible so that we can put those in as critical spawning habitat. Um, Personal interest stuff would be things like exactly when they hatch out and where they're going as juveniles and where they're spending those first 3 to 5 years before they go into go into their spawning mode. And then trying to figure out uh, how these fish are finding their post-spawning feeding areas when they're not going back to the estuaries and they're moving off into these lands. And the Anoka River is it would be a prime spot because it's basically a really slow moving lake, and that's just full of all kinds of invertebrates at the surface. I've been blown upstream in like a 5 mile an hour wind. It's a very interesting place.
It looked like on the microchemistry that the ones that did go to the estuary might stay there for more than 1 year, maybe two. It's hard to kind of tell by the scale, but is that true? Do you think they would?
So
Yeah.
so the coregonines, as far as we know, the coregonines can't deal with full marine water or those really cold temperatures below freezing. So they have to at least be in, you know, semi-fresh water during the coldest parts of the winter. So I think let me see if I can go back here real quick. Let me get on the right. So yeah, so there's that dip there right at the 4 mark, and that's that winter dark check. Um,
Hmm.
so so it and it's the actual concentration of strontium can't be directly correlated with because there's all kinds of other things, especially these fish are eating mollusks. So they could be getting some of that strontium from their marine derived nutrients rather than just absorbing it through the water in their gills. So there's a whole bunch of, you know, not really contradictory, but
Confounding things, maybe. Yeah.
yeah, confounding. Thank you.
Hmm.
Confounding issues with
Yeah.
this. So.
Okay. We have Randy on. I wonder if Randy wanted to
Can
make
you hear
any
me?
comments or thoughts. Yeah, we hear you.
Okay. I'm going to leave my my video off, but excuse me. But I think that Bill's right that they go down into the estuary and in the brackish water. I do think they can tolerate full salt for brief periods, so they can move along the coast a little bit. But during the winter, they would have to come into some sort of brackish water to survive it. Because under ice out there in the Bering Sea or the Chukchi, where the salinity is about 32 parts per thousand, the water underneath that is about minus 1.7 or so Celsius, and these guys can't go. Their fluid temperatures, uh, the fluids will freeze at about minus 0.6 or 0.7. So they're at least a full degree um, of freezing if they were to try to go out there. So they don't have the antifreeze proteins. But these broad whitefish are interesting because it does look like they go down and they might spend 2 or 3 years associated with the delta, and then they come up into freshwater and they go to spawn, and most of them don't go back again.
Right.
They live in freshwater then. So Bill had talked about that Chauvin Lake. Chauvin Lake was there was kind of a winter village there that used to go specifically to catch whitefish in the winter, and it's a deep enough lake that it's not it doesn't get plagued by low oxygen or anything, and it was a major food resource for people there in the Central Flats. But most of the lakes in the flats are too shallow, and they would probably go low oxygen in the winter. But that was a big one. And so it was interesting that that Bill was able to find that one in there and realize, oh yeah, they're living in the river, but also in that lake there.
Yeah.
When out in the Selawik area, I did a telemetry program with broad whitefish, trying to see where they spawned, and some of them went up the Kobuk River, uh, and we had 60 some transmitters out that lasted for about 3 years each, and most of them didn't leave Selawik Lake. And I think they spawned in the lake. It's a
Oh,
big lake. I think it's 30 kilometers by 15 kilometers, but
yeah.
its maximum depth is about, I think it's about 5 meters. And but there are these wind-blown areas on the sides that get good gravel for spawning, and most of our broad whitefish were out there for 3 years. They would come into the Selawik, and that's a freshwater lake. And then they would come up into the lakes around the Selawik mouth and smaller lakes and feed. Um, but we were thinking they're spawning out there in the lake. But big lakes are really hard to survey with for transmitters. Yeah, you can
Or
see the
Sellewick Lake, yeah.
Sellewick Lake, and
Yeah, it's the third largest lake in the state.
yeah, yeah, big. And but we haven't figured out how to go out and sample them in the winter. It's a really challenging piece of water there.
That's the one that's about 5 meters average depth.
Yeah.
No, it's five
Yeah,
meters maximum depth. It's
Maximum.
much shallower in a lot of
yeah.
places.
There are lots of places where it's, 2 or 3 meters for, 3 or 4 miles along the edges and stuff. So, yeah.
Yeah,
Thanks, Reed. Good to hear from you. Yeah.
that's nice, Bill. Yeah, thank you.
Thanks. Um,
You see Dylan Unland's message in the
yeah, I
chat?
see that.
Yeah.
So a lot of the stuff that we learned from traditional knowledge is their basic movements and where to catch them, because that's what fishermen are really good at, is knowing where to catch them. And then we can use some of these more advanced techniques like radio telemetry or microchemistry and stuff, and try to piece together, a larger life history. But yeah, we have one full-time staff person in Sellewick, and he's our maintenance guide. So when we have other outside researchers come, we send Sonny with them, and he's their bear guard and boat driver, and takes them to all the places that we don't know, because he grew up on the land, and he knows it like the back of his hand, doesn't need a GPS. And we couldn't do a lot of our work without people like that cooperating or being our employees. And that's the same for pretty much all the refuges have one or several, depending on how large they are, local hires just for that purpose.
Yeah, it's a great question, Dylan. Something I think we're trying to ask more and more of throughout fisheries, I hope. How to think about indigenous knowledge. So, and that subsistence fishery that you described, Bill, is that like pretty much, are there peak times of it, or is it pretty more consistent over the summer and fall?
So they, depending on what they want, what the fishermen are really want. So like this section of, so the village is here, and this is one of the spawning grounds. This is the Fish River, one of the spawning grounds that Randy found in his telemetry work. And their humpback whitefish and least ciscos spawn in that area. And that's they'll target that particular, the lower part of that particular river if they want eggs, because dried eggs, they make they use dried whitefish, or they use whitefish eggs, whitefish flesh, and caribou fat to make, and a lot of sugar to make something called Eskimo ice cream. And it doesn't taste like what you would think it tastes like with those ingredients. And lots of berries too. They put ockpicks and blueberries and stuff like that in it. So it's a really interesting mess. So the part of the reason why the village of Sellewick happens to be the largest of the 11 villages in this region, outside of Kotzebue, because Kotzebue is the airport hub, is because they can consistently put a net in the water any time of year and catch fish. They trade dried pike with Point Hope for muktuk, which is whale skin and blubber, for dried pike. And most people would think dried pike, but yeah, if you're a connoisseur, it's dried pike and seal oil. And you don't get cold during the winter eating a diet like that.
Thanks. Very interesting. Any other questions out there for Bill? Oh, let's see. There's one from Jason Leppy. You can read that as well. Basically, building some habitat suitability models to maybe predict other areas that have similar substrate or flow based on the spawning grounds that you found.
Yeah, so another grad project. So there's a big permafrost thaw slump that's out here on the in the Selawik, and the spawning grounds for sheefish is just down below it. And hopefully Ray Hander, who's another one of our whitefish colleagues in the Fairbanks office there, might present on it in the future. But so before that, we did a mark recapture project um, on sheefish up there, and there was a grad project done trying to bring together a whole bunch of remote sensing and on the ground stuff to try to figure out why they were spawning in that particular area. Because there's another large, very similar, the Tagawik, which we shorten to the tag, runs this way, and then the Cougar Rock River runs out here. But the tag would seem to be a very likely spawning ground. But none of the locals would ever go there to catch sheefish. When we had this big slump, we ended up having some out-of-state hunters up in that area, and they caught sheefish, and everybody said, well, they're lying. And then I went up there and radio tagged sheefish a couple of years later. And yeah, we found sheefish up there, and it was probably because the Selawik, which is usually a tea-stained, tundra-influenced system, was running like the Yukon. It was just chocolate milk, heavy turbidity. So some fish probably went up there to figure it out, but we haven't ever been able to find fish. That was in 2006 and 7, and we weren't able to find sheefish up there again. So those suitability models, there's something else involved rather than what we could manage to pull in on that sheefish project. So difficult question to answer.
We answered it well, and that's a good question to think about. But yeah, the nuances we still don't understand to build strong predictive models, it sounds like. Yeah. All right. Well, thanks again, Bill, for volunteering to step up. This was a great follow-on to what Randy taught many of us about InCanoe, especially those of us more focused in the Great Lakes. Just fascinating to hear more about all the coregonids up there in a very different region. So hope everyone can join us March 20th, I believe. It'll be a great talk by Alex Gatch looking into the potential importance of transport mortality as an potential impediment to our reintroduction of bloater, Coregonus hoyi, into Lake Ontario. So we look forward to that talk in about 6 weeks. Until then, everybody have a great weekend. Thank you.